We've been doing at the Cornell Lab of Ornithology. At the lab, as Bronwyn mentioned, I'm currently a science editor working with the Birds of the World project. Before that, I was a postdoc working in the Fuller Evolutionary Biology program, which is housed at the Lab of Ornithology. And I'll be presenting some of the research that I've done as a part of that work. While I'm interested in most aspects of birds and many aspects of nature in general, I'm especially interested from a research perspective in trying to understand how some of that amazing diversity has evolved in the first place. But before I get started, I wanted to first take a moment to acknowledge my collaborators on this research, in particular, Dr. Jen Walsh, who has led the Oriole project since I left my postdoc position back in 2019. So this has really become her project since then, as well as Dr. Bronwyn Butcher, Dr. Vanya Rower, and Dr. Irby Lovett. And there's been many other people also involved in this work. So to start, my specific research interests center on the process of speciation, or how and why new species evolve. The most common model to explain how new species evolve is through a process called allopatric speciation, which involves some ancestral population, say this green circle, getting split into two or more populations. These populations are usually separated by some barrier. That might be a mountain range that slowly rose up, a glacier that came down, or a river. While these populations are separated, they're unable to interbreed and they start to evolve and accumulate differences, whether that's due to adaptations to the slightly different environments where they now live, or some random genetic mutations that have accumulated over time. And over time, those barriers that initially divided them may disappear, allowing them to meet once again. And there are a range of possible outcomes that may happen once they come into contact again. The two populations may not have evolved very many differences, and they may interbreed freely and fuse back into a single population. Or the two populations may have evolved so many differences that they don't interact at all and are totally distinct species. Or the third option, they may be mostly different, but still able to exchange at least some genes. And when this happens, a hybrid zone may form. And at least from my perspective, as someone who's interested in speciation, this is one of the most exciting outcomes. So hybrid zones are interesting to me and to a lot of people because they represent cases where these two populations have evolved some differences, but they're still capable of interbreeding and exchanging genes. But despite forming hybrids, the two populations usually remain distinct. And it's the differences, the ones that really prevent them from completely fusing back into a single population, that can really help us to understand what is important in speciation in the first place, and what can cause species to evolve. In addition to learning about how and why species have evolved, another aspect of hybrid zones that fascinates me is that they're constantly changing. They are an inherently dynamic system. The two populations or species, in most cases, in a hybrid zone are constantly interacting not only with each other, but with other species, with their environment, which itself is constantly changing. But one problem that we have when we study hybrid zones is that we're usually limited to studying them from a single point in time, which is, kind of a challenge when we're studying an inherently dynamic system. But sometimes we're lucky and we've been able to look at hybrid zones over a prolonged period of time. And there have been some really interesting observations and some very important insights into what actually maintains and contributes to hybrid zones in these long-term studies. For example, in a hybrid zone between black-capped and Carolina chickadees are moving northward as winter temperatures get warmer. While black-capped chickadees can survive in warmer temperatures, they're being forced northward too because Carolina chickadees are the dominant competitor. And by studying this hybrid zone over multiple points in time, researchers were able to learn that climate and behavior both play a key role in how these species interact and hybridize. And that interest in speciation and change over time influenced my PhD research at the University of Wyoming, where I studied hybridization in sapsuckers. Two species, the red-breasted sapsucker on the left and the red-naped sapsucker on the right interbreed and hybridize regularly in an area of California and Oregon where they produce offspring with a mix of traits from both species. And what was really interesting and exciting in this case was that not only were these species hybridizing, but similar to the chickadee example, the area where they were hybridizing was changing very fast. And you can see this change when comparing museum specimens collected from the same place 40 years apart, where there used to be a mix of plumages representing both red-naped and red-breasted sapsuckers in the 1970s over here. You can see birds with black and red on the chest. But 40 years later, only red-breasted sapsucker plumage remains. So all these birds have only red on the breast on the right. And using an integrative approach that included data from museum specimens, genetics, and climate data, I set out to determine what led to these rapid changes as a way to understand how differences between these species evolved and is maintained over time. And in the sapsuckers, I ended up finding that climate may have been important in driving the movement of the hybrid zone, but that other factors such as mate choice may have also been important in maintaining aspects of the system. After my PhD research, I shifted to studying hybrid zones in the Great Plains, including the one between the Baltimore and Bullock's Orioles, which I'll be mostly talking about today. The hybrid zone between these two species was very well studied between the 1950s and the 1970s, and it really contributed greatly to our early understanding of hybrid zones in general. Like many others, this particular hybrid zone was thought to have formed after these two species diverged in separate refugia during the Ice Age, and then they expanded their respective ranges after the glaciers receded and came into secondary contact kind of in the middle of where their ranges would be, and where they now interbreed and form hybrids. While we learned a lot about the Oriole hybrid zone through these early studies in the 60s and 70s, much of the research focused on describing patterns of hybridization. They didn't really get at understanding the actual mechanisms driving hybridization. In addition, because of these extensive historical studies, though, we actually have an incredible baseline to compare to if we want to revisit these locations, which we did. So a little brief overview of some of the early history of research in the Oriole hybrid zone. The early interpretations of hybridization in the 1950s and 60s led Baltimore and Bullock's Orioles to be officially lumped as a single species called the northern oriole. The researchers argued that the presence of extensive genetic mixing, along with what was then considered a wide hybrid zone, led to this opinion. Work on thermal tolerance differences a little bit later led to a reconsideration of this approach, and the northern oriole was once again recognized as two distinct species. However, some of these changes were also based on changes in just how we think about hybridization and hybrid zones in general. How we interpret the original data from the 50s and 60s is very different from how researchers thought about it back then. And a big part of that are the decades of additional research on hybrid zones in general have shown us that even with relatively high amounts of hybridization, selection can still be really strong and act to maintain two distinct species. But in those early days, only the amount of hybridization was considered, with very little thought of what was acting as a selection pressure. All right. So with that in mind, we have this incredible baseline. We can now ask, has the Oriole hybrid zone changed over the last 50 years? And by understanding how the hybrid zone has or has not changed, we can begin to better understand what mechanisms may be driving the patterns of hybridization between the two species. So as I've mentioned, the Orioles meet and hybridize in the Great Plains of North America. And not only do the Orioles hybridize there, but in total 5 pairs of species or subspecies of bird hybridize there. Some of my other research involved comparing how the hybrid zones of different species are similar and different from each other to explore whether there are shared aspects of climate or something else that could be affecting how and why biodiversity is maintained on a large geographic scale. but that's a topic for another talk. So I'd be happy to come back and talk about that. But here I'll just be focusing on the Orioles. And for the Orioles and actually all the rest of the taxa that hybridize, rivers that run from west to east act as corridors that connect western populations to eastern ones, providing narrow bands of forest or woodland habitat that cut through the seas of grass, allowing species like orioles or flickers or buntings that require trees to expand their ranges. Most of our research on the Oriole hybrid zone focuses on one particular river system, the Platte River, which flows from west to east across the Great Plains through Colorado and Nebraska before flowing into the Missouri River. Orioles also hybridize along several other rivers that crisscross the Great Plains, but the Platte River is one of the best studied transects of the hybrid zone, and so we focused our efforts there. And along the Platte River, we have this wonderful riparian corridor of dense shrub cover and trees that allow birds like orioles to spread across the Great Plains, effectively connecting deciduous eastern forests with western forests of the Rocky Mountains. Across the Oriole hybrid zone, males are very different in their plumage. As you know, in Maryland, Baltimore Orioles in the upper right-hand corner here show extensive black heads with narrow white wing bars versus the Bullock's Orioles in the upper left-hand corner, which have bright orange faces and a bold orange supercilium and bold white wing panels. Hybrids, these three birds on the bottom here, can be very variable, often showing variable amounts of black and orange in the face. This middle bird is kind of your average hybrid. But depending on the precise ancestry of that hybrid, they can look either more like a Bullock's Oriole with just a little bit more black in the face or around the eyes, or they can look more like a Baltimore Oriole, sometimes with just a narrow orange supercilium that cuts through the otherwise all black head. Early researchers of the hybrid zone used this variability to estimate ancestry of hybrids, and they developed a scoring system based on the assessment of all the different plumage patches that differ between the two species. And we can still use the same scoring system to assess levels of hybridization, but we can now supplement it with genetic data to provide even more accurate assessments of ancestry. As part of ongoing research on the hybrid zones in the Great Plains, researchers at the Lab of Ornithology revisited many of the same sites where researchers from Cornell back in the 50s and 60s studied hybridization, and they resampled the Oriole and other hybrid zones here. So we've used many different methods to study hybridization and change in the Great Plains. And one approach that I've been really interested in is to see how the Orioles have adapted to climate and how that affects where they overlap and hybridize. So using museum data and data from citizen science platforms like eBird, as well as climate data from the 1950s and from now, we can actually map areas of overlap between the species and see how that has changed over more than 50 years. So this allows us to ask two main questions regarding climate and the hybrid zones. One, does climate maintain the position of the Oriole hybrid zone? And how has climate change affected the Oriole hybrid zone over time? To go about addressing those two questions and to understand how climate and climate change has influenced hybridization in the Great Plains, I constructed climate niche models based on observations in localities of hybrid individuals. So in this case, I'm only looking at birds that were classified as hybrids to build these models. So briefly, this process basically involves extracting climate data from each location, putting it into a model, and then determining where those preferred climate conditions actually occur on the landscape. And so we can end up with a final product that looks something like this, where it's a predicted map of where the hybrid zone should occur. And so to zoom in on the Oriole hybrid zone predicted map, basically this map is a probability of occurrence, where the whites and the pinks and the yellows and oranges represent a low chance of hybrids occurring, or put another way, areas that are unsuitable for hybridization to occur. The green represents a high chance of hybrids occurring there, or it's a place where hybridization is likely to occur. To make these maps simpler, I turn them into a simple presence-absence map. So basically, any area that had a more than 70% chance of having hybrids, I cut out. And it basically creates a nice little range map for the hybrid zone. And then what I can do is I can compare the historical location of the hybrid zone to the current location of the hybrid zone. And by plotting these two, we can see that they look a little different. The current niche occurs further west than the historical niche. But what do these shifts actually mean? There can be a number of reasons independent of climate change that could lead to changes in where a hybrid zone occurs on the landscape. To see whether these changes are the result of the hybrid zone actually tracking changes in climate over time, we can take the set of historical climate conditions where the hybrid zone was found in the 1950s and 60s and see where on the landscape those conditions occur now. And what we get is this orange blob. And while it's not a perfect fit, we do see a lot of overlap in this orange and between these orange and green areas. And this tells us that the Oriole hybrid zone has at least partially tracked changes in climate over time, and that it may be at least partially maintained by climate. We can look at this change in another way too. We can look at a single climate variable that was important in our model and see what range of values the birds occurred at. And for the Oriole hybrid zone, we can see that the current and historical niches are very similar in these two cases. So it's further evidence that they're responding somehow to climate, and that climate may be important. So we can revisit our first questions about climate and hybridization. And so does climate maintain the Oriole hybrid zone, and how has climate change affected it? Well, we do indeed find evidence that climate is at least partially responsible for the maintenance of the hybrid zone in terms of where it is located. But as I mentioned earlier, hybrid zones are complex and dynamic, and there could be other factors that are important in this system. Just like in the chickadees, where climate addressed one half of why it was moving, but behavior and the interactions between the species explained more nuance in how that hybrid zone was changing. Well, luckily we have plenty of other opportunities to investigate this hybrid zone. Another way that we've studied the Oriole system is with genetic data. So using samples that our team collected from along the Platte River transect across Nebraska and Colorado, as well as some samples from the American Museum of Natural History, the University of Wyoming, and the University of Washington, we ended up with 316 samples of Orioles, and we were able to generate genetic data. And we used a couple of different methods to generate our genetic data set. As a first step, we used a next generation sequencing approach called DDRAD. And what this basically does is it uses enzymes to chop up the genome into tiny fragments, and then we sequence those tiny fragments. We then look for any regions within those fragments where the two species are different, as a way to assess how genes are exchanged across the hybrid zone. And we ended up with about 1,900 of these different regions, which are called single nucleotide polymorphisms, or SNPs for short. And we use these to assess genetic ancestry of each individual to see how genes flow across the hybrid zones, how genes are exchanged between the two species. So what does the genetic structure of the Oriole hybrid zone tell us about selection here? What's going on genetically in these two species and their hybrids? So here's what's called a structure plot, which I'll walk you through step by step. What this essentially shows is the genetic ancestry of an individual, or the proportion of an individual's alleles that come from either one species or the other. So as we move from left to right across this figure, each vertical bar represents an individual, with the two colors indicating the proportion of their genetic ancestry that comes from one species or another. So in this case, yellow represents Bullock's Oriole ancestry, and red represents Baltimore Oriole ancestry. The two ends of the plot, so all the way on the left and all the way on the right, represent populations that are pure, or individuals that were sampled well outside of the hybrid zone. So in this case, Baltimore Orioles that came from New York and Bullock's Orioles that came from Washington, about as far from the hybrid zone as you can get. And those give us a baseline to understand what's going on where the two are mixing. So using these proportions, we can then calculate what's called a hybrid index for each individual. And what that measures is basically the proportion of ancestry that comes from one particular species. In this case, we measured our hybrid index as the proportion of Bullock's Oriole ancestry. So a value of 1 here means it's a pure Bullock's Oriole, and the value of 0 means it's a pure Baltimore Oriole. And everything else in between, so 0.5 is a perfectly intermediate hybrid. A 0.8 is a bird that has mostly Bullock's Oriole genetic ancestry, but still has some Baltimore Oriole ancestry, and so on. And as you see, as we move from west to east, the amount of Bullock's Oriole ancestry in yellow declines. And we can simplify that by just plotting the average hybrid index from each location in the hybrid zone. So where the width of these boxes here is just the amount of variation in ancestry at each location. And so here we can see that as we move from pure Bullock's Orioles over here, we still have mostly Bullock's Oriole genetic ancestry from areas in Colorado. And then we hit this area of transition where we get mixed ancestry between Baltimore and Bullock's Orioles. So there's a lot of hybrids in this area. And then once we move beyond that, we get into mostly Baltimore Oriole ancestry. And we can also plot these ancestry values directly based on their geographic position. And we can fit a mathematical Klein or a formula to that transition to better describe the shape of the hybrid zone. So again, in this case, 0 down here represents Baltimore Oriole ancestry, and 1 up here is Bullock's Oriole ancestry. And we see that same pattern of a very abrupt transition from Bullock's Oriole ancestry, and then this narrow area of transition, and then Baltimore Oriole ancestry. And what this means is when there is a very steep area of transition, it usually indicates that there's strong selection against hybridization, or strong selection for one species to mate with itself. And this area of steep transition occurs in northeastern Colorado. Yet another way that we can look at the genetic data is by plotting the frequency of the different ancestry values. So basically, this plot pools all of the individuals from across the hybrid zone. There's no geography here. And from this plot, we can see that birds that have genetic ancestry similar to either pure Baltimore Orioles or pure Bullock's Orioles are dominant in the hybrid zone. And there is actually very few purely intermediate individuals. And this type of distribution of individuals suggests that, again, there's very strong selection against hybrids, or strong selection for individuals to mate with their own species, which is called assortative mating. So this is all getting really cool, but we can go some steps further. So these are not Orioles, but I'm using them as to illustrate a point. So a common assumption of hybrids is that birds that look more like one parent or the other have more genetic ancestry from that parent, while birds that look perfectly intermediate between the two parents have very mixed genetic ancestry. However, that's not actually always the case. Sometimes selection of a certain trait or behavior found in one species or the other can lead to that specific trait to spread throughout a population faster than the rest of the genetic background, such that the phenotype or the outward appearance of an individual does not match their genetic ancestry. And this can lead to some very wrong assumptions about a hybrid zone if you're only using the outward appearance of an individual to understand what's happening. A famous example of this in birds is that of the white-collared and golden-collared manakin hybrid zone in Panama. To the east, over here, are genetically pure golden-collared manakins, and to the west, over here, are genetically pure white-collared manakins. And in the middle, and here, are hybrids. And phenotypically, most of the hybrids look very much like golden-collared manakins. But genetically, especially over here at sites 4 and 5, they're actually almost genetically pure white-collared manakins. So in this case, a preference by females for the yellow collar led it to sweep through this entire population much faster than the rest of the golden collared manakin genetic background, leading to some very interesting patterns. And it tells us a lot about selection in the system and what's kind of driving hybridization here. In the case of the Orioles, however, we do indeed find a very strong correlation between genetic ancestry and their outward appearance. It's almost a one-to-one relationship. So birds that do look more like Baltimore Orioles have more genetic ancestry from Baltimore Orioles and vice versa. And while this isn't hugely surprising, what this means is that we can now directly compare our genetic results from our modern samples of the hybrid zone to the characterization of the hybrid zone using just phenotype from the 1950s and 60s. And in doing so, we have additional lines of evidence that the hybrid zone has not only shifted westward, but the width of the hybrid zone has also gotten narrower over time, suggesting that selection against hybrids or selection for assortative mating has gotten stronger over time. So over a period of 50 to 60 years, the hybrid zone actually appears to have shifted between 40 and 70 kilometers to the west. So that's like a kilometer per year, which is pretty fast in this kind of system. And based on some of our previous results looking at climate, some of that could be driven by climate change, but there could be many other factors that are at play here. So to wrap up kind of some of the complex genetic stuff, this is a scary figure, I know. It hurts my brain to look at it too much, but so far the genetic analyses that I've talked about, while they sample across what's effectively the entire genome of a bird, we only use 1,900 markers. And I say only in quotes because 25 years ago, that would have been a preposterous number that would have cost millions and millions of dollars to create. But now 1,900 markers is kind of a trifle, and we can go much further. And we did dive much deeper into the genetics of the Oriole hybrid zone. In 2023, Jen Walsh led a paper that had us sequence the genome of a bunch of Orioles, I think 55 Orioles from across the hybrid zone, to truly understand the underlying genes and the genetic processes that could be contributing to reproductive isolation in the face of hybridization. So using a whole genome resequencing approach, we generated a data set of 11.6 million genetic markers for 55 individuals. And then we can compare the levels of divergence or levels of difference across all of those 11.6 million markers to get an idea of what genes, the actual genes, might be contributing to speciation or reproductive isolation between these species, and where in the genome these differences are. And when we do this, we get this scary figure, and it's called a Manhattan plot. And it is so named because some people think it's reminiscent of skyscrapers in a city skyline. I don't get it. They just look like a bunch of dots to me, but they're skyscrapers to some people. Each dot is one genetic marker, and they're plotted based on their physical location in the genome. So basically what chromosome they're on. And so on the bottom here, I have labeled what chromosome each region is. And so they're kind of broken up. Gray here is 1, black is 1AB, gray here is 2, and so on and so forth. And so they're plotted based on their position on the genome and their level of divergence. So here on the left, FST is a measure of genetic divergence, 0 being no difference between Baltimore and Bullock's Orioles, and 1 being a fixed difference. So it's always different between Baltimore and Bullock's Orioles. And we have some really high areas of genetic difference between these species. And yeah, so there are several very high peaks indicating that those are genes that are highly divergent or highly different between the two species, with those genes potentially encoding important differences that help to maintain reproductive isolation or contribute to reduced hybrid fitness in the Orioles. In addition to several high peaks that we see throughout the genome, we see all the way on the right over here. Sorry, I'm making sure there. We see the kind of baseline level of divergence is much higher on the Z chromosome. And so the Z chromosome in birds is one of the sex chromosomes in birds. In birds, the sex chromosomes are called Z and W, as opposed to X and Y in mammals. And unlike in mammals too, it's the males that have 2 copies of the Z chromosome, whereas females have a Z and a W. So this elevated region of divergence on the Z chromosome is very significant. Only about 0.4% of all the genetic markers across most of the genome show an elevated level of divergence. But on the Z chromosome alone, there's over 9% of the markers show elevated divergence. And what's especially significant about that region is that it likely represents a chromosomal inversion. And so this is basically an area that on the chromosome that's flipped around. So you just take a chunk of the chromosome and flip it in space, and you get an inversion. So all the genes are arranged in the opposite direction than they should be. And this structural difference means that there's reduced recombination, which can make it easier for divergence and differences to accumulate without the risk of the coevolution of certain genes to be broken up. It makes it easier for adaptive changes to evolve. So in this case, many of the differences that we were able to identify in the Oriole system relate to melanogenesis genes. And so melanogenesis genes basically encode for pigment, and black pigment in this case. And so that's one of the main plumage differences between male Baltimore and Bullock's Orioles. And so the identification of a number of genes that encode for the deposition of black pigments could be a significant difference between the two species and could contribute to the plumage differences that we see. In addition to the genes that could encode differences in plumage, genes that are linked to eye development and eye photoreceptors were also identified as different, which, if they're linked to the plumage differences, could help to explain the evolution of visual preferences and the evolution of mate choice between these two species. Furthermore, across many different groups, not just in the Orioles, but across many different taxa, inversions on sex chromosomes have been linked to divergence between hybridizing species, such that inversions on sex chromosomes in general may be very effective barriers to reproduction and may be particularly important in the early stages of speciation. So with all these additional data, we can also revisit our geographic clines from earlier, adding more lines to that plot. The gray line, that's a little hard to see here. That's our original line that we plotted earlier from our first batch of genetic data. And on top of that, we can now add a cline for the inversion on the Z chromosome, as well as a cline of the melanogenesis linked genes. And we see that there's a lot of overlap there, suggesting that all of these factors may be working concordantly in shaping the hybrid zone. And the fact that most of these clines have such a similar shape and occur in such a similar area of the hybrid zone suggests that selection is strongly acting in this hybrid zone. But as we can see from this blue line, that is a bit shifted to the east, all that selection may not be acting in the same direction. I mean, hybrid zones, as I mentioned earlier, they're messy. Biology is messy. And there's no single answer to these kinds of questions. But in the Oriole hybrid zone, many different factors are likely working, perhaps in concert with each other, but perhaps sometimes not. And it's the combination of all these things together that give us what we now know as Baltimore and Bullock's Orioles. So to wrap things up, we used a combination of historical museum specimens, climate data, and genetics to understand patterns of speciation and the mechanisms contributing to the maintenance of the Oriole hybrid zone. The Oriole hybrid zone appears to be experiencing strong selection. It seems to be tracking changes in climate, and there's a relatively steep geographic climb in kind of the genetic mixing of the two species. We find similar shapes, similar cline shapes and locations across different types of genetic markers. We found what's a putative inversion on the Z chromosome that is likely contributing to reproductive isolation in the system. And many of the genes that we identified as different between the two species are involved in melanogenesis, which could contribute to plumage differences. And lastly, a bimodal distribution of hybrid index values or a bimodal distribution of genetic ancestry suggests that, again, there's very strong selection against hybrids in this system. And so what do all these results tell us about Orioles? Despite ongoing hybridization, a combination of extrinsic or outside forces like climate and intrinsic or internal forces like genetics are likely contributing to the maintenance of these two distinct species that we know and love. Even relatively small reductions in hybrid fitness can lead to very large, long-term consequences that ultimately lead to the evolution of distinct species. And speciation, as we've seen with the Orioles and other systems, can be the result of a complex and sometimes messy combination of factors that don't always work together, ranging from genetic incompatibilities to adaptation to different climate conditions, mate choice, different molt strategies, different migration strategies, and many other things. And to give you an idea of what a hybrid zone with no selection would look like, if we consider an average dispersal distance of 95 kilometers, which comes from, in this case, a red-winged blackbird, but it's the closest thing to an oriole that had an estimate for dispersal distance. If there is truly random mating with no selection, then the hybrid zone would be about 2,600 kilometers wide, or 8 times wider than our current estimate of hybrid zone width. So we have really strong evidence in that case that there is indeed selection maintaining these two species. So in the case of Orioles, we have good evidence in at least climate adaptations and possibly genetic incompatibilities arising from a Z chromosome inversion could be contributing to the further evolution of these species, with further evidence for genetic differences in eye photoreceptor, cell development, and retina morphogenesis, which could result in differences in how the species perceive each other and their plumage differences. So I'd like to thank the various funding sources that we had for this project, our sources of data, including the Cornell Museum of Vertebrates, the University of Wyoming Museum of Vertebrates, the American Museum of Natural History, and the Burke Museum of Natural History, and various many people who helped with analyses. And all the lovely artwork that you saw throughout the talk was courtesy of Megan Bishop. And I would be very happy to take any questions from anyone.
All right, thank you, Sean. That was wonderful. I'm going to ask you to unshare. I'm going to put a spotlight on you.
Yeah.
And then we'll open it up for questions. You can put your question in the chat or raise your hand, and I can call on you to ask a question. I have a basic question, Sean.
Yeah.
Are the hybrid individuals, are they sexually viable? Do they?
Yes.
Do they reproduce?
Yeah, they are fertile. And in fact, in most cases, we don't actually see a lot of hybrid sterility in birds in general. It's an interesting kind of peculiarity of hybridization in birds. We don't see that same level of sterility that we see in mammals, for example. And there are many hypotheses as to why that is. One being that pre-mating barriers, so mate choice, like behavior differences, song differences, plumage differences are all more important in kind of the evolution of species than genetic incompatibilities. But there are many hypotheses for that.
So would the would the hybrids mate with each other, or would they choose either a Bullock or an Oriole, Baltimore?
It varies, and so that's where you get that gradient in ancestry. So a 50-50 mix often is the result of one Baltimore Oriole and one Bullock's Oriole mating and producing offspring. But everything in between could be either the result of hybrids mating with each other or back crossing with one of the other parents. And so we get this gradient in different levels of hybrid or genetic ancestry. Because yes, hybrids are very viable. But even though they're viable, there's probably something about them that leads them to be less fit than the pure parents. And that could be they might not live as long, they might lay fewer eggs, they might have a harder time finding mates because they look different or sing a weird song. They might migrate to the wrong place. All these little things add up, and over evolutionary time, even those small like reductions in fitness, if they're producing slightly fewer offspring over time, it's going to eventually be kind of an evolutionary dead end.
Interesting. Okay. Any questions?
Let's
I have
see.
more, but I'm going to open it up to the group.
I see a question in the chat. What was the vertical label on the FST graph? So yeah, the vertical label FST it's a measure of genetic divergence or genetic difference. So a value of 0 is there is no genetic difference at that genetic marker between Baltimore and Bullock's Orioles. And oh yeah.
Sean, is there a common ancestor that's been noted for the New World Orioles?
I don't remember what the Oriole. I'm having a a hard time remembering where in the phylogeny the Orioles are. So they're in the blackbird family. And I don't remember which group of blackbirds they're most closely related to. One thing I did not mention, which is also interesting with the Baltimore and Bullock's Orioles, they're not actually each other's closest relative. And that's very unusual to find a hybrid zone between two species that aren't each other's closest relative. Usually hybrid zones form between sister species. Um, the the closest relative of each Baltimore and Bullock's Orioles occur in Mexico. Um, and I think the sister species for Bullock's Oriole is a species called black-backed oriole. And I can't remember what Baltimore Oriole is most closely related to. But the high levels of genetic divergence, especially that we see on the Z chromosome, could in part be the fact because they're not each other's closest relatives. So we see especially high levels of genetic divergence compared to other hybrid zones that we study.
And how do the food sources compare and contrast between the two?
I think they're pretty similar. I alluded to it earlier in the talk, but one of the earlier studies that eventually led to the northern oriole being resplit into Baltimore and Bullock's oriole was a study on the physiology differences between the two species. And so they actually found that Bullock's Orioles were more able to cope with hot, dry conditions compared to Baltimore Orioles. And so there is very strong direct evidence that they were better adapted for their specific environments. And so over evolutionary time, a Bullock's Oriole wouldn't be, extending east into the eastern deciduous forest, and Baltimore Orioles wouldn't be extending west into the dry, like arid pine forests of the Rocky Mountains.
There's a question in the chat from Mike about the Z chromosome has significant recombination. I assume, therefore, that it is a chromosome with good size, unlike our Y chromosome. Is that accurate?
Yes, yeah. So the Z chromosome in birds would be like equivalent to the X chromosome in mammals, and the W chromosome would be the small one equivalent to the Y chromosome.
Okay. Any other questions? And I love that you used museum specimens for your research, again, highlighting the importance of these wonderful assets that need to be protected and preserved, because we can go back in and ask new questions to
Yeah,
these old.
who would have thought in the 1950s we would be able to do what we do now with these specimens? I see another question.
Al has his hand up. Go ahead, Al.
Hi, very interesting. I got lost in on some of it, but it's fascinating. I put up early in there, when you first showed sort of where the hybrid zone was at, it sort of reminded me of like a tornado alley kind of a image there. And I actually put that in the chat. And then I looked up, that was from like actually up until the 1990s, and they're now showing the tornado alley is actually shifting further east from where
Huh,
it used to be. Um,
interesting.
but then
Which
my
is,
standard question that I've asked everyone on these Thursday learnings is, how did you get interested in this from when you were a child in school, or where'd your interest come from, and however far you want to take us through that evolution of your interest?
yeah. Yeah, I mean, I've always been interested in nature and kind of outdoors things, and I think my spark bird was an American oyster catcher. Um, I think that was when I was 6 or 7, and I've kind of been obsessed with birds ever since. Um, Yeah, and then in college, I kind of started working on in a genetics lab, and um, yeah, I kind of just got really fascinated with understanding how and why the diversity of species that we have evolved, and I got really interested in like looking at hybrid zones because they just allow us to directly investigate like what are the key things, what are the key factors that that might contribute to why species evolve.
Yeah, so this
I
may
love,
be metaphor, but we've had such a variety of responses to that. From one was like someone went to a random table, at a choice of choosing a course, so different evolution of the scientists that have presented
Yes.
here.
yeah, I love the fact that I didn't know that, I hadn't heard that term before. My spark bird, what sparked
Hmm.
my interest in getting into birding. I love that. I'm gonna start asking that.
Yeah,
Mike
my
had a,
original oh, I was gonna say my original idea for my PhD project was to look at hybridization in oyster catchers, but that just would have been too challenging.
Mike, did you want to talk about, you followed up with the comment about bullets preferring hot, dry surprises me.
Oh,
I would have assumed they prefer colder climates because of the mountains to the west.
yeah. So I mean, they do occur in cooler regions, but then they also occur in like lower elevations in Arizona and New Mexico, and California. So they also occur in some very hot and dry um conditions. I think in particular, probably the dry aspect is might be the more important part of that. But it might go hand in hand with with hot as well.
And now you piqued my interest. Why would the oyster catcher have been a more complex?
Oh, well, it just would have involved I mean, certainly compared to sapsuckers, it would have involved travel to other countries and dealing with permits in other countries and for research, and much harder to get a big sample size of oyster catchers.
Thank you.
All right. Any other questions for Dr. Billerman from Big Cornell Lab? Well, thank you so much for coming
Oh,
and
thank you so
sharing
much for
your,
coming and inviting me. I had fun.
yeah, this was great. And thank you all for coming, and stay well, stay curious, stay outside, and keep looking for those Orioles and hoping that they continue their winning streak. I hope I didn't jinx them now. I hope I didn't jinx them. But good night, everybody. We'll see you next time for the wonderful world of sand.
Good
It's
night.
always something different. It's always something
Yeah.
different.